Okay, this video is the slavery metaphor for dementia from the Rogers theory of dementia. It's the supply and demand theory of dementia. That's me, Pete Rogers. Okay, so here is a normal looking brain on CT scan for a middle-aged person. It's a relatively tight brain, meaning that the cerebral cortex means bark, like the bark of a tree. If you have a tree stump, the outer periphery is the bark. So the outer periphery of the brain is where the gray matter ribbon is, where the cell bodies are, and it's up tight against the inner table of the skull. This is a normal middle-aged brain. Okay, now here is a demented brain. There's an increased amount of cerebral spinal fluid along the outer surface of the brain. So this brain is atrophic. The brain is shrunken. This is what I see in the vast majority of cases. One of the things I do is I'm a neuroradiologist. I look at demented brains every day. And the most characteristic finding of a demented brain is that it's severely atrophic. You'll sometimes see multiple infarcts. You'll sometimes see multiple periventricular small silent infarcts are sometimes called. You sometimes see a big cortical infarct. Yeah, that's true. But the vast majority are diffuse atrophic brain with a little bit of mild ischemic disease, but not enough to explain their dementia. Okay, so now we're going to get into the slavery metaphor to help you make sense of it. Okay, so by the way, when I hear the word slavery, everybody thinks black people. Okay, my father's from Ireland, okay? The Irish have been slaves of the British for 1,000 years, okay? And even currently now, they're trying to, side the Irish, and it's bad, and it's happening. But anyways, I just want to share with you, we're going to talk about slaves, in particular a slave here being whipped. That's going to be relevant to our metaphor for the brain cell. So this idea of Irish being slaves, that goes back for over 1,000 years, okay? So now we're going to kind of get into what does that mean. One other situation, it's much less common, but there have been examples of Polish women owning Irish slaves, including my ex-wife, pictured here. Okay, it's not her, but that looks a lot like her. All right, so anyways, here is a cell, a brain cell, a neuron, and the key point is that when there's elevated cytoplasm calcium, that will cause activation of the cell to do whatever it normally does. So for a brain cell, this will cause activation of the brain cell to release its neurotransmitter, which most often in the cerebral cortex is glutamate. 90% of cerebral cortex neurotransmitters are glutamate. It's an excitatory neurotransmitter. Excitatory means it causes an increased activation of the cell that it communicates with the postsynaptic neuron. The connection between two brain cells is called a synapse. This is the electrical system of the cell through these membrane ion pumps. We'll talk about that more in a moment, but it all comes down to regulation of calcium. Now here is a brain cell. So the first brain cell is called the presynaptic neuron. The synapse is the space between the pre and the postsynaptic neuron. So NT stands for neurotransmitter. When this presynaptic neuron is activated, its neurotransmitter goes to the synaptic cleft, diffuses across the cleft, and exerts an effect on the postsynaptic neuron. The mitochondria, you'll see quite a few of them up here in the cell body region where the DNA is with the nucleus. There's also mitochondria down here in the axon terminal where the neurotransmitter vesicles are stored. The key thing to activate release of the neurotransmitter is an increase in cytoplasm calcium. That is like the aristocratic ion. Sodium and potassium, magnesium, they're minimum wage workers. They just do what they're told. Calcium is the boss ion. When it comes in, it causes the cell to be activated and the neurotransmitters to be released. So you have to know that. Calcium is the big deal. All right, so here will be a neuron. And by the way, EMF, electromotive force, as may be caused by Wi-Fi or holding your cell phone to your head, will open up these voltage-gated calcium channels, so it has the effect of being a mild stimulant upon your brain cells. And the longer you hold it next to your head, the worse that is. Okay, so here's a glutamate neurotransmitter being released across the synaptic cleft, activating the postsynaptic neuron. There's other things that can activate the postsynaptic neuron receptors for that neurotransmitter. One of them is GP, glyphosate, that has the effect of an excitotoxin. It's thought that what it does is the glyphosate means glycine phosphate, essentially. It then can substitute for glycine in binding this receptor on the postsynaptic neuron. So it functions like a glycine activator. Okay, that allows more calcium into the postsynaptic cell. Aspartate, as in aspartame from the sweetener, that can activate this receptor to allow more calcium into the postsynaptic cell. What I'm doing here is giving you examples of stimulants, because people will often have multiple simultaneous stimulants adding up to cause more activation of the postsynaptic neuron. Okay, beta amyloid protein will also increase activation of the NMDA receptor for glutamate. And that gets elevated when you eat a high-fat diet. The high-fat diet causes insulin resistance, like in diabetes. The elevated insulin then has to be cleared from the blood. The enzyme to clear the insulin from the blood is called IDE, as an insulin-degrading enzyme. It has a high affinity for insulin to remove it, but it is only made in limited quantities, the insulin-degrading enzyme. Its other function is to clear beta-amyloid protein from the peripheral blood. When you have a shortage of insulin-degrading enzyme because it's used up with the high amounts of insulin in the blood due to insulin resistance and diabetes, then you get elevated beta-amyloid protein, which travels from the periphery of the body to the brain, and it exerts an effect on this NMDA receptor, getting it to be more excitable, to open up more, so more calcium gets into that postsynaptic neuron. The bottom line is it acts as a stimulant. So there's 3 extra stimulants right there: glyphosate, beta-amyloid protein, and aspartate from aspartame. Okay, caffeine is also a stimulant for this receptor. Well, it acts indirectly. Same thing with excessive psychological stress or sleep deprivation. All of them are going to increase glutamate activity. That's going to be highly relevant to why people get brain damage. Okay, in order to prevent brain damage, the cell has to pump the calcium out of the cell, out of the cytoplasm. Normally, the extracellular matrix calcium is about 10,000 to 15,000 times higher in concentration. Whenever you pump against a high concentration gradient, you must use energy to do that. So you need ATP, adenosine triphosphate. You can pump it out through the knockout exchanger. You can pump it out through the plasma membrane calcium ATPase. You can also pump it into the endoplasmic reticulum of the presynaptic neuron. Just get the calcium out of the cell. Okay, but you must use ATP. So the point is you need lots of ATP. So pumping that calcium out when it's present in high amounts is like having a boat with a hole in it and the ship is sinking, and you've got to pump that water out fast or the ship will sink. That is what happens to your neurons. This is a very good metaphor for what is happening to that neuron. You must get the water out of the boat before the boat sinks. You must get the calcium out of the cytoplasm before the neuron is exhausted or it will die. Now, in addition, I meant to stay on that previous slide. Here is the mitochondrial inner mitochondrial membrane, and these are the electron transport proteins, and several of them are proton pumps. They pump protons into the intramembranous space. That's the space between the inner mitochondrial membrane and the outer mitochondrial membrane, which isn't in this picture. But the point is, I'm showing you is, in all my med school, college biochemistry books, and I own a bunch of them, there was nothing about mitochondrial inhibition. I knew there had to be a lot of them because I ran across a few of them in reading literature papers, so I decided to make a dedicated effort to find them. I quickly found over 60 of them, and many of them are very common, very, very, very common. You ever heard of Tylenol and metformin and selective serotonin reuptake inhibitors and propofol and statins and fluor, hydroxynonenal from omega-6, atrazine sprayed on corn, glyphosate sprayed on soy, etc. Okay, so anyways, what was my theory? My theory was basically you have a baseline metabolic rate in a neuron, and that is normally matched to the supply and demand of the glucose-oxygen delivery. I jokingly abbreviated glucose-oxygen delivery as GOD, not capitalized, of course. So basically, in order for a neuron to survive, it must be able to meet its metabolic demands. It needs the glucose and oxygen to convert that into ATP for energy, so it'll have the pumping power to get the calcium out of its cytoplasm. So if you drop glucose and oxygen delivery because you're eating a high-fat diet, causing blood sludge, which drops oxygen delivery, drops tissue perfusion, and then you also have a lot of sodium to vasoconstrictor, which drops the blood supply to the brain, you've got a problem, okay? And caffeine's bad because caffeine is a stimulant increasing metabolic rate, while it's simultaneously a vasoconstrictor like sodium, dropping blood flow to the frontal lobes of the brain. So you can see how it's a double screw job. It increases metabolic rate in the neurons, but drops blood flow because of vasoconstriction to the brain, about 15-20%. It's a significant amount. Okay, so then what else is going on? We talked about a lot of these other things having a stimulant effect. Caffeine, psychological stress, sleep deprivation, monosodium glutamate, manufactured free glutamate, beta-amyloid protein, glyphosate, attention deficit meds, amphetamines, tobacco, etc. Okay, that's a lot of stimulants. And then guess what? There's another little secret here we haven't really got into much, but it's all those mitochondria inhibitors. I showed you there's a whole bunch of them. And now I'm going to show you the slavery metaphor that will make sense of it, because people sometimes have trouble with this. Imagine there is an unfortunate slave, which that is not me. I realize it might look a little bit like me, but that is not me. I'm much more skinny and muscular than this individual. Okay? And that is not my girlfriend either. It may look a little like her, but that is not her. Okay? So anyways, brain cell normally matches its supply and demand. I previously called this theory the neurovascular uncoupling theory. Normally they're coupled, the amount of blood supply and the metabolic demand. So supply, again, is oxygen and glucose delivery. Demand is the metabolic rate of the neuron, which relates to the amount of intracellular calcium it has in its cytoplasm, which activates neurotransmitter release. Then the energy producing capacity of the mitochondria. You need the mitochondria to make ATP. That's where most energy in a cell is made. Okay, the next issue is what are the stimulants involved? Because the more stimulants and the more powerful they are, the more hyperactive that neuron becomes. That can also cause anxiety. It can cause insomnia in people. A lot of young people have insomnia and anxiety. Okay. Now, high-fat meals, we talked about that, dropping oxygen and glucose delivery. You also, when you tend to get leaky gut from these types of meals, and you get postprandial endotoxemia, meaning gut toxins from bacteria, LPS, lipopolysaccharide from gram-negative bacteria, LTA, lipoteichoic acid from gram-positive bacteria. Also, chronically, if you eat processed foods and a lot of meat, you get elevated bodily iron, manifested quite often as blood, high levels of serum ferritin, but you'll have increased amounts of free iron in the blood, which you don't want. You want iron is like fire in a house. You want iron in the fireplace to heat the house. You want iron in the stove to cook your food, but you don't want it anywhere else or it breaks stuff. That's what iron is like in the human body. And when you start having excess bodily amounts of iron, and most men are iron overload by the time they're 30 in a Western country. Most women are iron overload by the time they're 60 in a Western country. Then you're going to have increased amounts of oxidative stress that are prothrombotic and pro-inflammatory, okay, which predispose to blood clotting and dropping nutrient delivery to your brain cells. Okay, we talked about sodium being a vasoconstrictor, netosis and clotting. That's a little bit like amyloid clotting, but we'll talk about that some other time. Okay, lots of mitochondria inhibitors. So what does this all mean? Let me explain. So the stimulants are like telling your slave, run faster. You tell the slave, you must run faster, and he gets whipped if he doesn't run fast enough, okay? Then despite telling the slave to run faster, you go, well, you know what? We don't have a lot of extra food. We're going to starve the slave. So the slave is still told that he must run faster, but simultaneously you're starving him. It's a little bit difficult for the slave to maintain his ability to run fast if you starve him. And if that wasn't enough, this lady's a little bit sadistic. She takes her whip and she starts whipping the slave on its legs. Run faster, slave, while she whips his legs. The little red spots on his legs are to indicate bleeding. So you can see the slave will try to hang in there, but if you're whipping him in the legs and, tearing up his legs with the whip, and you're starving him, depleting him of energy, how is the slave going to keep running faster? Well, that is exactly how Westerners routinely treat their brain cells. And eventually what happens is the slave dies. So here is a statue of a slave dying. Actually, it looks like a slave dying. This is really a famous statue of the dying Gaul, perhaps a victim of Julius Caesar's minions. But anyways, the slave will die because it simply cannot meet its metabolic demands because it's deprived of nutrients, oxygen and glucose, and simultaneously its mitochondria are inhibited, so the slave just dies. And that means the brain cells just die. And they die in the millions and eventually the billions, and the person becomes demented. And that's the Peter Rogers, MD, theory of neurodegeneration due to supply and demand.